Hi class, it's Mrs. Baraboo, and today's video is going to be on our M&M Lab. I want to be able to do a demonstration for you so you understand what you're going to do when you walk into class next. So the lab supplies that you're going to need is a cup of M&Ms. Your M&Ms will be all different colors. And you will need a plate that has an E on it for experiment. And you will need another plate that has a D on it for discard. And that's the supplies that you're going to need. Please do not forget your lab write-up. You should make sure that you read every single page and you know exactly what you're going to need to do next class before you even walk into class. Okay, so I'm going to do the demonstration of the Part 1 lab, exponential growth. The M&Ms are representing cancer cells. So we are going to investigate how cancer cells grow in the first experiment. So the first thing you're going to need to do is to take your M&Ms and put them on the D plate for discard. And then if you're reading the directions, it says place 2 M&Ms in a cup. So that's your cup here. We're going to put 2 M&Ms in the cup. And then it says, so that's your trial 0 that's already located on your table, that you have 2 M&Ms or 2 cancer cells. You're going to shake the cup, and when you dump it out, please make sure that you're very careful to keep the M&Ms on the plate, because these are the M&Ms that you're going to want to eat later, and you don't want it to impact your lab results. So we're going to pour them out onto the experiment plate, the E plate. And then it says, for every M&M with the M showing, add another M&M and record the new population. So if you get a close look of the plate, there's actually both of them have an M showing. M is facing up for both of them. So I'm going to take 2 M&Ms and I'm going to add them to my cup. And I'm going to count 2 plus 2. And I'm going to record 4 in my trials, so if you could take a look at that. Okay, the table, I put a 4 in my trial number 1, and now I'm ready to perform the trial again. So I'm going to take the plate and I'm going to curl it up to make sure my M&Ms don't fall out, and I'm going to put them back into the cup. And now I have the 4 M&Ms in my cup. I'm going to shake it again, and I'm going to pour it out. So now this time there's 3 M's that are showing. So I'm going to add 3 into the cup, or 3 onto my experiment, and then I have to add 3 to my total. So if I had 4, when I add 3, now I'll have, hopefully you can do the math, that would be 7. Okay, so 7 M&Ms on my plate, 7 M&Ms recorded, and then I'm going to take my plate again and put them into the cup. Okay, it says repeat steps, so you repeat this part of the experiment until you are done with 15 trials, or if you run out of M&Ms. So you may use all the M&Ms, so your numbers should be getting fairly large, or after you have filled in the entire chart and there's no more room, then you're also done with that experiment. Okay, after you do part 1, you're actually not going to do the write-up part until you have done both experiments. So you're not going to graph your data yet. Once you get done with your table, you're going to turn to part 2, and that's M&M exponential decay. And if you can see, I put the 130 on there. Okay, the 130 is because I counted ahead of time before I did the video. There's 130 M&Ms in my cup. So what you're going to have to do is you're going to have to put all the M&Ms like they were to begin the first lab, and you're going to have to count every single M&M. And then how many M&Ms I gave you in the cup, you're going to record under trial 0. Once you've done that, all of your M&Ms go in the cup. This takes skill. You have to be careful that you get all the M&Ms in the cup so that your experiment's not flawed. Okay, you have all the M&Ms in the cup. This time when you shake the cup, you dump out the M&Ms, you remove the M's that are showing. So when you do that, you're going to have to kind of shake your plate so that all the M's are flat. Okay, can you see the ends are flat? And you have to be careful to look carefully for the ones that have ends and put them on your discard plate. So you can get a close-up of this. All the ones that are ends, I am taking off the plate. And you have to be careful because yours are not going to be pretty Valentine's Day colors. Yours are going to be all the different colors, and especially those yellow ones, you have to look very carefully. Now your partner should be able to help you with taking them off so that it's not going to take you as long as I am with doing it with one person. And again, when you're taking them off, you have to be careful that you're not turning the other M&Ms. And it will take you a while to do this. The good news is you're getting rid of quite a few, so that means each time it'll be easier for you to do it. That first time is going to take the longest. I'm almost done here. So after you get all the M's off, you can count them as you're doing, you can count them as you're moving and then subtract that number, because that's what you're taking away. Or once you have the plate of all the ones that are not M's, then you can go ahead and count them and put them in your cup. So I have 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, 28, 30, 32, 34, 36, 38, 40, 42, 44, 46, 48, 50, 53, 56, 59, 60, 63, 66, 67, 68, 69, 70. So then I'm going to record the 70 on my chart here. And as you can see, it's gone down pretty significantly. Okay? And then you would repeat that step. So what I would do from there is these are the cancer cells still. I have 70 cancer cells left. And the object of decaying, and think about that's what chemo is doing with people who have cancer, and we can remove those cells. And if you remove those cells, they can't multiply and grow exponentially. So you would remove those, and again, you would record how many are left in your experiment. And you would do that, and I want you to pay attention. It says, continue this process and fill in the table. You are done when you have completed 10 phases, or when your M&M population gets below 4. So if you record the number 3, or 2, or 1, you're done. Okay? You're never going to record the number 0, because unfortunately, you're never going to get down to 0. Okay? An example of that is I have a friend who gave birth to twins, and she had a cancer that she passed on while she was pregnant. She died of the cancer because it grew exponentially before they were able to find it. Her twins have 1 cancer cell that was passed to them. They go for 6-month checkups now every year, twice a year, to make sure that that cancer is not multiplying and not growing. But they're always going to have that 1 cancer cell. So they're cancer-free because it's not growing or impacting them, but they have the risk of having the cancer because at any point that cell can choose to divide, and then that'll start that growth process of that rare cancer that they have. So I hope this helps you understand that. Please read through the lab. I want to explain some of my expectations that I have for you. First of all, with the graphing, I want you to make sure you choose a proper scale. So you have to look at your data and choose the scale. When you are answering the questions, I want to make sure that you ask your partner and you're trying to work through it and reading the directions carefully. And that starts with reading them before you even walk into class. Then you have to follow the directions and go through the process and record, or basically go through the experiment by yourself. Then what's going to happen is you're going to end up with an equation that's called an exponential equation, like we've been talking about in class, that's based on your data. But we all know that there's human error. I forgot an M. Okay, one of the M&Ms fell on the floor. Okay, those type of things are going to happen in an experiment. We know that that's not truly, a perfect world. So down here, you can enter your data into a graphing calculator, and it will show you how to use the graphing calculator to come up with a better equation, or an equation that's going to look similar to yours. Okay, so this process is just it's not as accurate as maybe the graphing calculator is going to do. So you're going to go through that process on the graphing calculator, and that's using the technology. Then you're going to answer the questions based on comparing what you got in the lab compared to what you got on the graphing calculator. And then you'll do the same thing for the second lab. The very last thing is the conclusion, the lab discussion, the so what part. Okay, how is this significant with what we're learning in class? And how does this apply to real life? And what does the stuff that we saw in the experiment, what's going on and what does it represent? So when you fill out this part of the lab, you need to use complete sentences. If they say why or explain, those are keys that you need to have complete sentences. This will be a test grade, so it's going to be important that you put your effort into it, not only for you, but for your lab partners. Your lab papers will be graded individually, and there'll be a rubric that you will be handed out on exactly how you're going to be graded for the test. I will tell you that group participation in the actual performing the lab is a big part of that test grade. So you need to make sure you're taking it seriously and you're using all the time in class efficiently. If that means helping counting, somebody moves on to the next step while somebody finishes up one step, somebody's using the calculator while you're setting up the next lab, whatever you can do to make your time efficient and work together, that's the reason why you have a partner. Okay? Otherwise, I'd just make you all do it by yourselves. There is absolutely no eating of the M&Ms. So I know you guys. Okay? You are not putting the M&Ms anywhere near your mouth until the lab is completely done. I have looked at your lab paper and I've specifically given your group permission to split up the M&Ms. But then if there's two people, you can split them up on the plates, and that's your snack or your Valentine's gift from Mrs. Baraboo. Okay? Again, I will be around to help you, but you need to be able to read and follow the directions. That's part of the lab assignment. All right, thank you, and I look forward to seeing you in class.